2 minutes, madam.
Yes, sir, no problem.
Yeah, madam. Hello. Hello.
Hello, good morning, sir.
Morning. We can start now.
Okay, sir. I'll
Yes.
start.
Yeah, please.
Okay. Hello, everyone. Welcome to Tawheed Foundation for Allied Healthcare Sciences, Bangalore. Today's class is focused on an important topic in microbiology, which is growth and nutrition of bacteria. So, before starting, I would like to give an intro. Like understanding how bacteria obtain their nutrients and how they grow is fundamental in microbiology because it helps us comprehend their survival, reproduction, and behavior in different environments. This knowledge is crucial not only for academic purposes but also for practical applications such as controlling infectious diseases, developing antibiotics, and using beneficial bacteria in industries like fermentation and biotechnology. So, let's explore the essential processes of bacterial growth and nutrition to understand how these tiny organisms impact everything from human health to industrial applications and ecological balance. So let's begin with the introduction. Microorganisms require nutrients to survive, grow, and reproduce. Nutrients are broadly classified into two categories, which is macronutrients and micronutrients. These macronutrients, which are required in large quantities, and micronutrients, which are needed only in trace amounts but are essential for various cellular processes. Both types of nutrients are vital in maintaining cellular function and overall microbial metabolism. This diagrammatic representation of micro and macronutrients. These are nutrients required by microbes in large amounts, which is macronutrients, major nutrients, because they form the basic building blocks and play major roles in cellular structure and function. Example highlighted here in the diagram include carbon, fundamental component of all organic molecules, that is carbohydrate, proteins, lipids, and nucleic acids. Oxygen, it is an integral part of organic molecules essential for cellular function. Nitrogen, present in amino acid, nucleotides, and other vital biomolecules. And phosphorus, part of nucleic acid, ATP, and phospholipids. Additional ions such as potassium, calcium, magnesium, and iron are also shown as micronutrients, macronutrients, because they have important cellular roles like enzyme activation, stabilizing structures, and electron transport in metabolism. This micronutrient trace elements, these nutrients are necessary in small quantities but are crucial for the enzymatic and metabolic activities of bacteria. Other trace elements includes manganese, zinc, cobalt, molybdenum, nickel, copper. Their main roles are as cofactors in enzyme reactions, enabling catalytic activity, electron transfer, and other metabolic processes essential for bacterial growth. So the significance of this diagram is the clear differentiation shows that macronutrients are needed for building cell components and energy metabolism, while these micronutrients enable biochemical reactions necessary for living cells. So understanding this helps explain why bacteria must obtain a wide variety of nutrients from their environment to survive and replicate. This also impacts how culture media are formulated to provide these essential nutrients for laboratory growth of bacteria. Macronutrients, which is major element. The major macronutrients essential for bacteria include carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. These elements form the building blocks of the major organic molecules. Carbohydrate, lipids, proteins, and nucleic acids. Carbon act as the backbone of organic molecules, while hydrogen and oxygen are part of organic compound as well as water. Nitrogen is crucial for amino acids and nucleotides. Phosphorus is found in nucleic acids and energy molecules like ATP, and sulfur is part of some amino acids and vitamins, all contributing to the bacteria's metabolism and structure. Other macronutrients and functions. Besides the major elements, bacteria require additional macronutrients such as potassium, calcium, magnesium, and iron. For vital cellular functions, potassium ions are important for enzyme activation. Calcium ions plays a critical role as a component of bacterial endospores. Magnesiums act as a cofactor for many enzymatic reactions, and iron is essential in electron transport system during cellular respiration. These elements maintain structural stability and facilitate metabolic reactions essential for bacterial survival. Micronutrients. Trace elements like manganese, zinc, cobalt, molybdenum, nickel, copper, though needed in very small small amounts are indispensable for bacterial enzymatic functions. For example, zinc is often found in enzyme active sites, manganese catalyzes phosphate transfer reactions, and molybdenum is crucial for nitrogen fixation in certain bacteria. Although required in trace amounts, the absence of these elements can severely impair bacterial metabolism and growth. Requirement of carbon, hydrogen, oxygen, and electrons. Carbon forms the backbone of organic molecules that make up bacterial cells. Hydrogen and oxygen are present in all organic molecules and in water, which is vital for life. Electrons play a key role in energy generation through processes such as the electron transport chain and redox reaction. Electrons are required for biosynthetic processes, providing reducing power necessary for the synthesis of molecules critical to cell function. Requirement of nitrogen, phosphorus, and sulfur. Nitrogen is essential for constructing amino acid, nucleotides, and certain carbohydrates and lipids. Phosphorus is critical in nucleic acid, ATP, the energy currency of the cell, and phospholipids that form the cell membrane. Sulfur is incorporated into the amino acid, cysteine, and methionine, and in vitamins like biotin and thiamine, playing an important role in enzyme function and structural stability. This image consists of three parts: bacterial growth, bacterial growth to microbial nutrition. Here is a detailed explanation of each component I'll give. I'll start with bacterial growth curve. This graph shows the typical phases of bacterial growth in a closed system, that is batch culture, representing the number of viable cells plotted in a logarithmic scale against against time. So we'll start with lag phase. At the beginning, bacteria are introduced to a new environment. Cells are metabolically active, adapting to the new nutrients and synthesizing enzymes, but not dividing yet. The population size stays almost constant. Logarithmic, which is exponential growth phase, cell starts dividing at a constant and rapid rate. This phase represents balanced growth where the bacterial population doubles at a fixed interval called the generation time. Bacteria are most sensitive to antibiotics in this phase. Third is stationary phase. Growth slows down and stabilizes because nutrients here are get depleted and toxic waste accumulates. The rate of new cell formation equals the rate of cell death. Total viable cell number remains constant. Here secondary metabolites like antibiotics may be produced here. Last is death decline phase. Nutrient exhaustion and accumulation of toxic byproduct lead to cell death here. In death decline phase, so viable cell number decreases exponentially. The test tubes illustrate this progression by showing the changes in the number of viable cells, dark shaded compared to non-viable cells, black over time. Ion uptake mechanism. These diagrams illustrate how bacteria acquire iron, an essential micronutrient, from the environment. Iron in the environment primarily exists in the form of Fe3+, which is ferric iron. Fe3+, ferric iron, which is not easily accessible to bacteria. Here, one second. Here, SIDO4. These are specialized molecules secreted by bacteria that have a very high affinity for Fe3+. Binding The cytophore bind Fe3 ions in the environment, forming a cytophore ion complex. Receptor side, here this is receptor side. The complex binds to specific receptor sites on the bacterial cell membrane. After that, internalization, the Fe3 plus is then transported to the bacterial cell where it is used for critical biological functions like electron transport and enzyme activity. So this whole process ensures bacteria can scavenge iron efficiently even in environments where free iron concentration is very low. Third is microbial nutrition and cell metabolism. This schematic shows the fundamental metabolic processes inside a bacterial cell connected to microbial nutrition. This diagram is not clearly visible. I'll try to explain everything. First is anabolism, which is biosynthesis, part of metabolism where the cell synthesizes complex molecules from simpler ones requiring energy. Nutrients are the raw material for producing cellular components like protein, nucleic acids, and lipids. Catabolism, breakdown of chemical nutrients like sugar to release energy. This energy powers cellular activities including biosynthesis, transport, and motility. Energy flow. The arrows here indicate the flow of energy from nutrient breakdown, which is catabolism, supplying ATP and reducing power needed for anabolic reactions. Last is waste products. Metabolic processes generate waste such as acids or gases. Example, CO2, which are expelled from the cell. This diagram highlights the interdependence of nutrient uptake, energy production, and biosynthesis, all critical for microbial growth and survival. So, all three diagrams together, these components demonstrate how bacteria grow and thrive by efficiently utilizing nutrients, acquiring essential elements like iron, Fe3, which is shown here, and balancing energy production with biosynthesis in their metabolism. Here in this diagram, you can see these dotted lines indicate few or no cells, these dots viable cells, and this black colored shows non-viable cells. Nutritional types of microorganisms. So, microorganism can be classified based on their sources of carbon, carbon source, energy source, and electron source. So, photolithotropic autotrophs use carbon dioxide as their carbon source, light as their energy source, and inorganic electron donors. Example include purple sulfur bacteria and green green sulfur bacteria and cyanobacterium. Photoorganotropic heterotrophs use organic carbon as carbon source and light such as purple non-sulfur bacteria use electron organic source. Chemolithotropic autotrophs get energy from inorganic chemicals and carbon from carbon dioxide. Example, sulfur oxidizing bacteria and nitrifying bacteria. Chemolithotropic heterotrophs uses organic carbon, energy source inorganic chemicals, and electron source inorganic electron donor. Example says Begitoa species. Chemoorganotropic heterotrophs, these used organic carbon, same as carbon source, organic chemicals, same as energy source, and organic electron donors, same as carbon source. Example is most non-photosynthetic microbes, for example, Escherichia E. coli, fungi, and protozoa. Growth factors. So, certain organic compounds called growth factors cannot be synthesized by some bacteria and must be obtained from the environment. This includes amino acids, purines and pyrimidines, vitamins. For example, Enterococcus faecalis required 8 specific vitamins, Haemophilus influenza needs Hemi, and Mycoplasma required cholesterol. These growth factors are essential for various biosynthetic pathways and contribute to the rapid growth and survival of bacteria in nutrient limited environments. Nutritional types of bacteria. Bacteria are further classified into phototropes and chemotrophs based on their energy source. Phototropes use light energy, while chemotrophs harness energy from chemical compounds. On the basis of carbon source, autotrophs utilize inorganic carbon dioxide, while heterotrophs depends on organic carbon sources. Examples include cyanobacteria as photoautotrophs and E. coli as chemoorganoheterotrophs. This classification helps in understanding bacterial metabolism and ecological roles. Growth and multiplication of bacteria. Bacterial growth is defined as an increase in cell number and mass. The most common mode of reproduction is binary fission, which is a most common method where the DNA replicates, the cell elongates and forms a septum, dividing into two identical daughter cells. So, some bacteria like Hyphomicrobium multiply by budding, while others such as actinomycetes, these actinomycetes reproduce by fragmentation. Understanding different multiplication method is important for studying bacterial life cycles. Bacterial growth curve, which is batch culture, lag phase. In a closed system with no fresh nutrients, the bacterial growth curve shows distinct phases. The lag phase represents a period where cells are metabolically active but not dividing. Bacteria adapt to the new environment by synthesizing enzymes and essential molecules, preparing for division. The lag phase duration depends on factors like the physiological state of the inoculum and nutrient availability. Log phase, which is exponential phase. The log or exponential phase is marked by rapid bacterial cell divisions. Here, cell divides at a constant maximum rate, a process called balanced growth where cellular components are synthesized in proportion. Bacteria are metabolically active and sensitive to antibiotics during this phase. Primary metabolites such as amino acids and nucleotides are produced, which are essential for cell growth and division. Third is stationary phase. So, in the stationary phase, bacterial growth slows down and stabilizes as nutrients become scarce and toxic metabolites accumulate. The rate of bacterial growth equals the rate of cell death. So during this phase, secondary metabolites like antibiotics may be produced. Eventually, the death phase, which is decline phase, ensures where cells die exponentially due to nutrient exhaustion and waste accumulation. So here is a diagrammatic representation of all four stages: lag phase, log phase, stationary phase, and death phase. This diagram illustrates the bacterial growth curve in a closed system, which is batch culture, showing how the population of bacterial cell changes over time and identify four distinct phases of growth. So x-axis represent the progression of time during bacterial culture incubation. Y-axis, logarithm of cell count, represent the bacterial population size on a logarithmic scale, which allows visualization of exponential growth phases. So, phases of the bacterial growth curve will start with lag phase. At the start, bacteria are introduced in a new medium. Cells are metabolically active but do not divide immediately here. During this phase, bacteria synthesize enzymes, RNA, and necessary molecules to adapt to the new environment. The cell count remains relatively constant here. Second is log exponential phase. Bacteria begin rapid and constant rate cell division. Population size doubles at regular intervals, reflecting exponential growth. Cells are most metabolically active and sensitive to antibiotics is highest at this point. This phase is important for research and industrial application due to high growth rate. Primary metabolite essential for growth are produced during this log exponential phase. Third is stationary phase. Nutrients become depleted and toxic metabolites accumulate in this phase. The rate of bacterial cell death equals the rate of new cell formation. So the overall population size plateaus. Some bacteria may produce secondary metabolites like antibiotic during this phase. Last is death decline phase. Nutrient scarcity and waste toxicity increases in this phase. The number of dying cells exceeded the number of new cells formed. So bacterial population declines exponentially in this phase. Some cells may enter dormant states or form spores to survive in this phase. So importance of this curve is this growth curve helps microbiologists understand and measure bacterial growth dynamics. Knowing these phases assists in optimizing culture conditions for research, industrial processes, antibiotic production, and understanding infection progression. So the log phase is often targeted when testing antibiotic because cells are most susceptible. So this clear visualization of phases is fundamental in microbiology and forms the basis for practical applications involving bacterial cultivation and control. Culture medium based on consistency, liquid broth, nutrient broth, solid broth is nutrient agar, blood agar, semi-solid used for motility test. So culture methods for bacterial culture include this bacterial culture include the streak plate technique for isolating pure cultures by spreading a sample across solid agar with a loop. The pour plate method involves mixing diluted bacterial samples with molten agar before solidifying, allowing growth inside and on the agar surface. The spread plate method involves spreading samples on solid agar to grow colonies on the surface. Enrichment culture enhances growth of specific bacteria by providing special nutrients which can be crucial for isolating pathogens from mixed population. So culture medium. Based on composition, there is six media: simple media, complex media, synthetic defined media, enriched media, selective media, and differential media. One second. As I mentioned, culture method, streak plate method for pure culture isolation, spreading sample on solid media with loop, pour plate method, diluted sample mixed with molten agar, which grows colonies inside and on surface, spread plate method, sample spreaded on surface of agar, enrichment culture enhances growth of desired organism by providing special nutrients. Last is aerobic culture medium. So here, aerobic bacteria require oxygen for metabolism and growth for and reproduce mainly by binary fission. The growth in batch culture follows four phases, which is again log log phase, stationary phase, and death phase. So various culture media and method exist to support bacterial growth and isolation. Aerobic bacteria require oxygen supplied through specialized culture techniques, which is shake culture method, surface culture method, and aerator fermenters. Shake culture method increases shaking aeration. Surface culture method increases growth on agar, slants, petri plates exposed to air, etc. Aerated fermenters, it is used for large scale cultures, especially in industrial use. Summary. So, bacteria require macronutrients and micronutrients for growth and reproduce mainly by binary fission. Their growth in batch culture follows four phases: lag phase, log phase, stationary phase, and death phase. So, various culture media and method exist to support bacterial growth and isolation. Aerobic bacteria require oxygen supplied through specialized culture techniques. So, questioning part. Hello, sir, am I visible? Sir, you are on mute
Yeah,
mode.
yeah,
Yes.
please, madam.
Okay. So, questions for 3 mark questions. There is 8 questions. Define macronutrients with examples. So, here what you can do, explain that macronutrients are elements required by microbes in large amounts. You can give examples like carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, etc. Second is differentiate between macronutrients and micronutrients. So here what you can do, mention macronutrients required in large amounts from cell structure and energy metabolism. Micronutrients is trace elements required in very small amounts, mostly enzyme cofactors, but are essential. Give examples of both each included macronutrients and micronutrients. Third is write any two functions of potassium and magnesium in microbial cell. You can mention potassium activates enzymes, maintains osmotic balances, and magnesium cofactors for many enzymes, stabilizes ribosomes and membranes. Fourth is name 3 micronutrients and their biological role. Here you can give examples: zinc, magnesiums, and molybdenum. Zinc, their biological role is zinc and enzyme active site. Magnesium helps phosphorus transfer catalysis. Molybdenum is for nitrogen fixation. Fifth, what is the role of phosphorus in microorganism? So phosphorus is vital for nucleic acid, DNA, RNA, ATP for energy transfer, and phospholipids of cell membrane. Six, state two requirements for microbiological growth apart from nutrients. So here you can tell temperature suitable for growth, educate oxygen or appropriate atmosphere. Also mention pH and moisture required for the microbial growth. What are growth factors? Give examples. So growth factors are organic compounds microbes cannot synthesize and must obtain externally from the environment. Examples you can give: amino acid, vitamins like B complex, hemi, which I mentioned earlier. Eighth, name the four phases of the microbial growth. So four phases of microbial growth is nothing but lag phase, log phase, stationary phase, and death phase. Elaborate everything in detail for microbial growth of four phases. 5 mark question.
Yeah, very nice presentation, madam.
So 5 mark
So,
question is pending.
five marks. Okay, done.
5 mark question. First, explain the role of carbon, hydrogen, oxygen, and electrons in microbial growth. Here, carbon is backbone of all molecules. Hydrogen and oxygen, components of organic molecule. With water is very essential for the life of bacteria. Electrons involved in energy generation, electron transport chain, and biosynthesis, which is reducing power. Second is describe the role of nitrogen, phosphorus, and sulfur in microbial metabolism. Here, nitrogen required for amino acid and nucleic acid synthesis. Phosphorus involved in ATP production, nucleic acid, membrane, and phospholipids. Lastly, sulfur, important component for some amino acids, cysteine and methionine, and vitamins involved in enzymatic function. Third, differentiate between primary and secondary metabolites with examples. So, primary metabolites produce during active growth, log phase, needed for growth, amino acid, nucleotides. Secondary metabolites, you can mention stationary phase, not directly involved in growth, but can provide advantages, for example antibiotics like penicillin, etc. Fourth, draw and explain the microbial growth curve. Here you can draw the curve showing all the four phases: microbial growth curve, log phase, lag phase, stationary and death phase. Explain each phases in terms of bacterial activity, population change and environmental factors influencing growth. Fifth is discuss different nutritional types of microorganism with examples. So classification based on carbon energy and electron source, photoautotrophs, chemoautotrophs, heteroautotrophs. Give examples of each such as cyanobacteria as photoautotrophs, E. coli, chemoorganoheterotrophs. Sixth is explain the importance of growth factors in microbial nutrition with examples. So growth factors are essential for organic compounds required by some microbes that cannot be synthesized them. They aid in critical biosynthetic pathway, ensuring survival and rapid growth. You can include examples Enterococcus faecalis and for cholesterol mycoplasma. All this importance of growth factors you can include in microbial nutrition. Um,
Over, madam.
over, sir.
Yeah.
Hmm.
So very nice class, madam,
Thank you,
on
sir.
bacterial growth and nutrition.
Yes, sir.
And we nicely explained about lag, log,
Love
stationary
is.
phase, and decline phase
Yes,
of
sir. I try
bacterial
to
growth.
cover every topic.
Yeah, yeah, madam. You have covered very nicely. And coming to one thing, I think factors affecting bacterial growth, madam.
Yes, sir.
So, what are the factors affecting, yeah,
Sir,
growth
yes,
factors you have
yes,
mentioned here.
factors. Yes, yes, sir. Sir, all
So,
these.
growth in the sense, there are some factors like temperature, yeah,
Correct.
pH,
pH,
yeah,
temperature.
yeah, pH.
Yes, sir.
Those may be, I think, it may be,
Included here.
yes, it may be
Yes.
included in this
Yes, sir.
slide, I think so.
Yes.
So, I
Uh,
think we didn't
it
read.
was missing. Oh, okay,
No
okay, sir.
issue, no issue.
Uh,
Yeah.
yes, sir.
So, but it is very fantastic and excellent presentation, madam, whatever it may be. So,
Thank you.
nice, madam. Shall
Thank
we wind up, madam?
Yes, sir.
Yeah. Thank you, madam. Thank you so
Sir.
much, madam.
Thank you. Thank you.
Thank you, madam.
Thank you, sir.